So in this video, we're going to be taking a look inside eukaryotic cells. So eukaryotic cells are quite a bit larger than bacteria. Recall from the last video that bacteria are on average smaller than 1 micron, and eukaryotic cells are on average 10 to 100 microns. So these cells are 10 to 100 times larger than their bacterial counterparts. And this means that they have a large surface to volume ratio. If you thought about this as a balloon, this would be a very large balloon relative to maybe the size of a water balloon, right? So filled with air versus water. So quite a big surface to volume ratio. And so this causes a problem in the cell because when you think about chemical reactions occurring, these solutes or the substrates, they need to collide with one another. And if they're in this very big space, that can be problematic. And so organelles can help solve this problem. So organelles are membrane-bound compartments that break up the large cell volume into smaller regions. And this has some advantages because the organelles can contain specific enzymes, and this can increase the efficiency of chemical reactions in those regions. And you can also get organelles that perform specific functions, so that changes in the number of organelles or the type of organelles within a single cell can lead to cell specialization. So we're going to talk about the organelles within eukaryotic cells. So first, let's begin with some general features of eukaryotic cells. And I'm showing you here a cartoon of a generalized animal cell shown up at the top here and a generalized plant cell. Now, both of these cells, like bacteria, have a plasma membrane, and the plasma membrane is made up of a phospholipid bilayer that may have cholesterol or sterols in it, and there's also integral membrane proteins that pass through the plasma membrane. And this creates a semi-permeable barrier to the outside environment. And so this can help protect the cell, but also can bring in important nutrients. Inside the cell, there's a lot of fluid. This is the aqueous environment we've been talking about. And we call this the cytosol. This is the intercellular fluid that bathes the organelles. And this is different than the cytoplasm, and sometimes these words are used interchangeably. But what the cytoplasm refers to is really everything inside the cell. It's both the organelles and the cytosol. So when we're just referring to this aqueous environment, we want to use the term cytosol. So let's talk about the first organelle inside eukarya. And this is the cell really that is its distinguishing feature. Its organelle that's its distinguishing feature. This is the nucleus. And the nucleus is composed of a double membrane. So that means that there's two lipid bilayers that surround the nucleus. And we call this the nuclear envelope. And there's a lot of support and structural proteins and pores for things to go in and out. And we'll talk more about this next lecture. And so here's the cartoon up here. And then down below is the transmission electron micrograph of a nucleus. So you can see the double layer structure and you can see some pores. We'll talk about these. And then there's these lighter areas, and this contains lightly packed chromosomes. This is where our genetic information resides. And then we have some darker packed areas with more DNA. And so here's where our genetic material is found inside the cell. And then we also have this really dark region here called the nucleolus. And the nucleolus is the site where ribosomes are made. So recall from the last video that ribosomes are made up of ribosomal proteins and rRNA, and those are made and assembled within the nucleus and then exported out into the cytosol. So ribosomes are found in eukaryotic cells just like they were found in bacteria. And ribosomes are made up of two subunits and are made up of ribosomal proteins and rRNA. And the rRNA is what catalyzes protein synthesis. So these ribosomes are in the cytosol and they're associated with different organelles, and this is where protein synthesis is occurring. And so cells that, and we'll come back to this, but cells that need to make a lot of protein have to make a lot of ribosomes. The next structure I want to talk about is the rough endoplasmic reticulum. So here's our nucleus in this gray cell here, and continuous with the nuclear membrane is the rough endoplasmic reticulum. So it's shown here in purple. And we can zoom in on this and we can see that the rough endoplasmic reticulum is studded with ribosomes. And so proteins that need to be shipped to other organelles or embedded in the plasma membrane or secreted actually need to get made within the rough endoplasmic reticulum. And we'll talk in the lecture about how that happens, how protein synthesis occurs in these flattened stacks. The rough endoplasmic reticulum is also important for making membranes, like the plasma membrane. And so this is a very important structure. And again, if cells need to make a lot of protein, it means that they need a lot of rough endoplasmic reticulum. The smooth endoplasmic reticulum is also continuous with the nuclear membrane. The only difference between the rough and the smooth endoplasmic reticulum is that the smooth endoplasmic reticulum lacks ribosomes. It doesn't have any of those complexes associated with this organelle. This organelle is responsible for synthesizing lipids and carbohydrates, and also plays a really important role in detoxifying drugs and poisons. It also is a site that you'll see a lot of calcium ions are stored and concentrated in the smooth endoplasmic reticulum. And when we talk about signaling and how cells communicate with one another, we'll see what an important role these calcium ions play in relaying signals and helping cells communicate with each other. The next organelle that I'll talk about is the Golgi apparatus. And so here, shown in purple, is our Golgi apparatus. And you can see that it's pretty close to the rough endoplasmic reticulum. So again, we have our nucleus, and we have our rough endoplasmic reticulum, our smooth endoplasmic reticulum. And right by the rough endoplasmic reticulum is our Golgi apparatus. And this Golgi apparatus is made up of these flattened stacks we call cisternae. And proteins actually are going to travel through this in vesicles from this cis side, so the side closest to the endoplasmic reticulum, to the trans side, and this is the side that is closest to the plasma membrane. So again, here's the transmission electron micrograph, and it's showing our Golgi cisternae, which are these flattened stacks. And this side here, this is the side that faces the nucleus and the rough endoplasmic reticulum, and this is where proteins that need to be processed and sorted and shipped away, this is where they're going to enter into the Golgi apparatus. And then they're going to move through this Golgi apparatus to the trans site and then basically be shipped away either to other organelles or to the plasma membrane or secreted. And again, we're going to talk about this. But this whole system of the nucleus and the rough endoplasmic reticulum and the Golgi apparatus, and we'll talk about in a little bit, lysosomes, they're part of this system called the endomembrane system. And then this is really important for packaging proteins and shipping them off outside the cell. So lysosomes are really important structures for breaking things down. They're commonly thought as or referred to as the recycling centers of the cell. And so they're shown here, they're all over the cell as these orange structures or yellow structures. And inside the lysosome are very strong digestive enzymes. So each lysosome contains 40 different kinds of digestive enzymes. And so this organelle is really good at breaking down either ingested substances or old organelles, anything that needs to be taken apart and dismantled. So here you can see these structures are pretty dense, and because they're packed with enzymes, and there's also areas that are basically being digested and then become clear, as you can see in these two electron micrographs of lysosomes. So these lysosomes, they actually, and the enzymes inside them, they work best at a super low pH. And so the inside compartment of a lysosome has a pH of around 5, whereas the rest of the cytosol has a pH of around 7.4. And so there's a very important protein pump on the membrane of this lysosome, and what it does is it pumps protons into the lysosome to maintain that low pH, to make sure that those enzymes are at their optimal activity. The next organelle I'll talk about is specifically just in plants and fungal cells. So we don't tend to have these large vacuoles in our cells, and other animals don't as well. And so what vacuoles do is they digest and recycle macromolecules, and they play a really important role in the storage of water and ions. So in plants, a lot of water is stored in a central vacuole and then used for photosynthesis and transported elsewhere in the plant. And in seeds, the vacuoles are also filled with proteins. And in flowers, they may also contain pigments. So these vacuoles can be specialized at different parts of development of a plant. All cells, plants and animals and yeast and protists, contain peroxisomes. Peroxisomes detoxify and break down fatty acids, ethanol, and other compounds. And they do this by oxidizing them. So they can oxidize something like alcohol. And oxidation typically produces hydrogen peroxide. And we saw an example of this when we were looking at our turnip peroxidase assay. And so peroxidase in the peroxisomes neutralize that hydrogen peroxide and turn it into water. And so this plays a really important role in the cell in detoxifying things that could be toxic and harm other proteins and macromolecules in the cell. So again, here's our peroxisomes shown in yellow, and then here's the transmission electron micrograph showing a peroxisome membrane, the enzyme core, and the peroxisome lumen. So we've talked a little bit about these next two organelles, the mitochondria and the chloroplasts. The mitochondria is the site of cellular respiration. And so in our cells, it's the site of oxygen consumption. And that's going to be used to generate ATP. And so here, our mitochondria is a double membrane organelle. And here we have the outer membrane and the inner membrane space. And we have our cristae. These are these flattened structures. And this is where, within here in the matrix, is where the citric acid cycle takes place in pyruvate processing. And then out here in the inner membrane, right, is where we generate our proton gradient, and we use that to make ATP. But what we didn't talk about is that the mitochondria is also an important site for amino acid and vitamin synthesis. And it also plays a really important role in regulating cell death. So when you think about cell death, you might think that that sounds like a bad thing. But in multicellular organisms, cells become old and they need to be replaced, and they need to do that without essentially breaking apart. And so these cells can regulate their own cell death, and this is called apoptosis. And the mitochondria plays a very important role in signaling whether or not the cell should die. Mitochondria are found in animals, but they're also found in plants and protists. So even though plants are making their sugar, their own sugar, they're taking that sugar and they're metabolizing it in the mitochondria. The mitochondria also has its own DNA, its own genetic material. And in lab, you're actually going to sequence a mitochondrial gene called COX-1 to find out what bug species you have. But we'll come back to why this is significant in just a second after we finish talking about chloroplasts. And so here's our chloroplast. And this also is a double membrane organelle, and it's the site of photosynthesis. And as we talked about, we have here these thylakoid membranes, right? And if they're a stack of thylakoid membranes is called a granum. And this is where the light capturing reactions take place. We have photosystem 2 that makes ATP, and we have photosystem 1 that generates NADPH. And that's going to make the energy to fix carbon in the Calvin cycle. And that takes place in the stroma outside of those flattened stacks. The chloroplast is also really cool because it has its own DNA. And this gives support to this theory called the endosymbiosis theory. And that theory states, or basically thinks, that mitochondria and chloroplasts were once free-living bacteria, and that they were ingested by eukaryotic cells, and they were mutually beneficial, and so they became an organelle. And this is supported by, again, the fact that it has its own DNA. They also have their own ribosomes to make their own proteins. So these are pretty cool organelles. So those were general features of all eukaryotic cells, and I want to touch briefly on some special features of animal and plant cells. So animal cells typically produce and secrete proteins and other components that make up the extracellular matrix. We talked about one of these components being hyaluronic acid. And these proteins and other chemicals provide structural and biochemical support for surrounding cells. And we'll talk more about these components probably in a later lecture. Plants, as I mentioned, they have a central vacuole that's filled with water and ions. They also have chloroplasts, so that's unique about plant cells. And surrounding their plasma membrane is a cell wall, and this cell wall provides structure and rigidity to this cell, and it's made up of the carbohydrate cellulose. This is a polysaccharide that's made up of repeating beta glucose that are linked together via glycosidic linkages, and then those layers of cellulose are linked together with hydrogen bonds that form this very stiff structure that keeps the plant in shape, so long as there's plenty of water and a vacuole full of water. So I mentioned this before, but the structure of each cell component correlates with the function of that cell in multicellular organisms. And so this means that the size and number of different organelles can vary, and they usually correlate with the cell's specialized function. So if we zoom in here to this electron micrograph of a pancreatic cell, what a pancreatic cell is supposed to do is that it's supposed to export digestive enzymes. These are proteins that need to be secreted. And so since proteins need to be secreted, it means we need a lot of rough endoplasmic reticulum. This is in contrast to something like a cardiac muscle cell. So muscle cells need a lot of energy because every time they contract, they need ATP to power that contraction. And so if you zoom in on a single muscle cell, you'll see that it's filled with mitochondria. They need a lot of mitochondria to make that amount of ATP. And so in class, we'll get some practice kind of looking in a cell and seeing if we can determine what its specialized function is based on what the organelles look like in a transmission electron microscopy picture. So that's all that I have for this lecture, and then I'll follow up with a really brief lecture about the cytoskeleton.